is Trisha Sivam, and I am a sophomore at the University of Alabama at Birmingham, and I am currently studying genetics and genomic sciences with a minor in biology and in chemistry. And one thing that I want to do with my life is become a researcher that essentially progresses the development of treatments and procedures that can ultimately help cure illnesses and people of their diseases. One thing that really interested me while studying here was the topic of diabetes, which is what I actually helped do research here in the De Miguel lab. Growing up, all my grandparents had diabetes, and that puts me at significant risk for developing it as I grow older, which is why I had that initial interest for it. One thing I learned is that diabetes not only affects our blood sugar levels, but other organs in our body, like our kidneys. Diabetes can actually impair our kidney function and how our kidneys work at filtering our blood and toxins out of it. And then these toxins that get filtered out are found in our urine. We can actually test how effective or how functional our kidneys are by examining our urine. When our kidneys are really damaged, we'll see high levels of protein because when the kidneys are filtering our blood to remove waste or extra fluids, the damaged vessels will leak protein into our urine. Healthy kidneys don't remove protein from our blood because protein is used all around our body by different cells. We can figure out how much protein is in our urine by running this thing called a protein assay. First, a dilute urine sample is pipetted onto the microassay. Then an indicator solution, which here is our Quick Start Bradford dye. This indicator solution essentially changes colors when it interacts with the protein. So when there is a low amount of protein, the color remains brown, and if protein increases, it turns more and more blue. The plate then sits for 5 minutes so that the indicator has time to react and change color. I then put this plate into our microplate reader. This microplate reader uses spectroscopy, which here is the process of using light in order to determine how much light is being absorbed by the sample, which would vary depending on what color it portrays to be because of its protein concentration. That we have ran our plate through the plate reader, it will tell me what the approximate concentration for the urine samples are. Technology like this can help diagnose kidney damage in patients that could be a result of diabetes, high blood pressure, or other chronic diseases. In order to do a protein assay, we need a couple of things. So here are my standards, which are essentially samples that have a known amount of protein in it. And my standards go down from a high amount of protein to a low amount of protein, and even include a blank, which is just water. And so we put those at the beginning of our protein assay so that when we're loading samples from, let's say, like humans or rats, we have something to compare it to in order to determine what is the actual protein concentration. And then this is my finished protein assay, and as you can tell, at the top it's blue and at the bottom is brown. And a blue color means that there's a higher concentration, and a more brown color means that there's a lower concentration. So if somebody were to have protein in their urine, they would have a more blue color to it.